Hey there, Zachary here, also known as Adam on Discord. I'm the team lead for TRegulators, and let me take you through our hackathon journey of building an open source platform for B-cell and T-cell epitope prediction. First off, I want to thank everybody on our team who contributed throughout this awesome event. Now let's showcase what we've built together. We envision a future where allergies can be cured through a vaccine, focusing on tolerance induction. Our project centers on epitope research, understanding the likelihood of a peptide to activate B cells or T cells. In our analogy, B cells are knights, causing allergic reactions, while T cells, specifically Tregs, are the princess, promoting tolerance. Our goal was to find peptides that avoid triggering B cells while engaging Tregs to promote tolerance. So building prediction tools to identify these peptides became our mission. For B-cell epitopes, we were inspired by SEMA 2.0, a web platform using AI models like ESM2 for conformational epitope prediction. We wanted to expand on their approach by generating predictions with ESM3, as this new model was trained on the observed antibody-space dataset, which includes B-cell receptors. Since ESM3 can consider structural data like surface area and atomic coordinates, we can use one model for sequence and structure predictions. We chose not to predict N-glycosylation sites or direct epitope comparisons as they were beyond our scope. We first replicated SEMA 1D predictions. Our teammate Steven fine-tuned an ESM2 8M model to match SEMA's sequence-only input predictions. We then processed PDB-IDs to extract residues and calculate RSA, ASA, N-glycosylation, hydrophilicity using the Hopwood scale, B factor as a proxy for flexibility, and a base electrical charge for each amino. We faced challenges replicating the fine-tuning for ESM3, so predictions are still a work in progress. For T-cell epitopes, we drew inspiration from mixed TCR pred, which provided a dataset for TCR to peptide MHC interactions for viral and cancer epitopes. They tested leave one epitope out prediction, and we extended this concept to predict TCR recognition for new proteins. We trained a classifier using ESM3 embeddings for TCR CDR3 regions and target peptides to predict TCR recognition and identify potential epitopes. Props to our teammate Ender for leading the TCR prediction. We also added allele-specific peptide MHC binding affinity as shown by our current T-cell epitope prediction set. For binding affinity, we initially used the IEDB Tools API but faced restrictive rate limits. Thanks to our teammate Flowers' academic email, we accessed DTU Health Tech's NetMHC PAN tooling and ran it on EC2 to predict peptide MHC affinity. Our primary deliverable is an open source epitope prediction web platform that features predictions for conformational B, linear B, class 1 and class 2 T-cell epitopes, sequence and structure support with PDB-ID and chain selection, prediction method selection. It breaks down the protein into peptides for analysis. There's job history, sharing of jobs, the output for jobs has sortable data and data downloads. There's visualization for conformational B-cell epitopes, both for the sequence and for the structure. For class 1 and class 2 T cells, there's a modal view to view the data for each HLA allele. And on the input form, there's support to select the alleles for analysis.